Okay, Meredith, can you hear me?
Yes, I can.
Oh, Meredith, I can't hear you. Okay, Meredith, try that again. Say something to me.
Hello?
Okay, there, I heard you. All right, Meredith, do the slides move?
Yes, they are moving.
They're moving, terrific. Okay, we're gonna get started then.
Okay.
Well, greetings, sports fans. Oh, your homeworks are graded. And got your tests are graded, so I will get these back to you at some point. Lab tomorrow. We're running gels. You guys done these before? So I'm gonna get everything out, go into the corner and sit down and read a magazine. And if you have any questions, you can ask me, but you should be able to do it yourself. You should be able to load the cassette, load the gel. Oh, we're purple today, are we? Oh, okay. You should be able to, okay. You should be able to load everything, get it on the gel, run it. If you need help turning the power supply on and getting it set, I can do that for you. But everything else, you have seen it done, been there, done that, got the hat. So I should be able to do it. And then the silver staining, which is the kit is all available. You should be able to do that, right? So tomorrow, I will be more of a helper than I will an instructor. So you're the experts now, so go for it, yeah? Yeah. Just doing silver staining. And if the calmodulin came off the column where I think it did, it might give us a pretty band. So we'll see. All right. Oh, goodness, what's up with this? Okay, you're not working? Yeah. Yeah. What's up with, you're off. Oh, I hope the ball didn't go. All right, soon as this warms up and going. All right, so we are considering the fit of pyruvate, right? Pyruvate is made from glycolysis. If you are a, let's see, glucose. And how many ATP do you make? 2 net. And how many reducing molecules do you make? Two, what? Two boxcars, two NADHs. And you get two A's. So the easiest thing to imagine would be to take these reducing equivalents and just put them back on here. And that's one reaction, and that's catalyzed by lactate dehydrogenase. Lactate dehydrogenase will make lactic acid. If you do that, then you there's two of those. And that is a so-called homolactic fermentation. And what's fermentation? It's making ethanol, right? Well, sometimes. What is fermentation? All your ATP via substrate level phosphorylation. Substrate level phosphorylation. That's how you make all your ATP. If you make all your ATP via substrate level phosphorylation, you, as an organism, carry out the fermentation. Now, it turned off again. I'm supposed to do this. We may have a lecture on the blackboard today. Now, this reaction doesn't just occur in microorganisms. Yes, it occurs in streptococci. Yes, it occurs in leuconostoc. Yes, it occurs in lactobacillus. But it also occurs in your own muscles. So type 1 muscles, type 1 fast twitch muscles have lots of glycogen, very few mitochondria per cubic centimeter, very little myoglobin. They burn their candles at both ends, so to speak. So if Remington were to chase Greg with a hot poker, Greg says to himself, I'll bet you if I outrun him, because that chap's a distance guy, he's probably not a good sprinter. And I've got longer legs and I'm scared. And he runs as fast as he can. That 40-second dash will be made with his fast twitch muscle. Fast twitch muscle. But after a time, his fast twitch will load up with lactate. They won't work so well anymore. And then he'll have to go to the slower twitch muscles, of which there are two types. Those will work mainly through oxidative phosphorylation. By then, because Rem's a distance runner, would have caught up to him, but hopefully by then, the poker would have cooled off. In which case then, it's not a hot poker, it's just a poker. Not as useful. And this is a reaction. We showed you the delta G of this. It's quite negative. Okay, you're my witnesses. You're my witnesses. Can you please go tell Kathleen to call AV and tell them that we have a dancing projector, a narcoleptic projector that, yeah. Well, I got plenty of chalk. Now, what do you do if you're yeast? You get excited, right? So, do baker's yeast make lactate? No, they make ethanol. How do they do it? Oh. So you still run glycolysis. You still make pyruvate. Now your next, you're going to decarboxylate this. Okay. Sure.
They're down to 180 person over there because everybody is out in quarantine.
Oh, great. Well, you know what? I'll just use the blackboard. Yeah. They probably won't get here till hell freezes over, but yeah. Sorry, forget my French. I just. Now, the enzyme that will be used is called pyruvate decarboxylase. This is a enzyme that is a homotetramer, and it contains a thymine pyrophosphate, sorry, TPP.
Dr.
Thymine
Baranovich,
pyrophosphate, yeah?
if you're not going to use this because the projector's not working, could you possibly stop slide sharing and point the camera at the blackboard so that Jess and I can see?
Shine the camera at the blackboard. Easier
If it's
said
at the
than
other one,
done.
that's fine. If it's at the other one over there, that's fine. But if you like use the one right behind you.
Okay, I'm gonna try something here. Oh, you gotta set that device up. Well, I don't have time for that. So let's see.
If you can't make it work, that's fine.
Well, this all doesn't reach that far, so I'm kind of out of luck on that one.
Yeah, that's okay.
Then okay, give that a try. Wait, to share though, don't you need to be a host?
No, it's just through my camera.
Oh, okay. All right, then yeah, that works. Okay, this will produce all right, who's this? Who's that? Aldehyde and carbon dioxide. Now, you have two NADH to make up. Remember, there's two of these. Make two of these, then dump those NADH here. And you reduce this, you make, and this is called alcohol dehydrogenase. Alcohol dehydrogenase, or ADH. A heavily studied enzyme. AH is the oligomeric enzyme. It lines up and it makes these big oligomers. It absorbs enormous amounts of product, substrate, excuse me, kicks out all kinds of product. Works very fast. And it's a zinc contained enzyme. Requires zinc and hexachloroethanol. Now, Are there other ways to make ethanol in bacteria? The answer is yes. And to illustrate that, we'll turn it around. That's in yeast. But other bacteria make ethanol too, but they do it in a different way. So in E. coli and a few other bacteria too, they have an enzyme when oxygen levels get low that's called pyruvate formate lyase. Pyruvate formate lyase. Pyruvate formate lyase. So this will take coenzyme A plus the pyruvates, well actually no, just as it to one pyruvate, and it will take the acetate part. This actually releases the form of a formic acid. So yet coenzyme A plus mate. So the formate, then that's an end product. Now you can also take pyruvate, and if you make this acetyl coenzyme A can have a couple of other fates as well, besides making formate. Here's what else you can do with it. What do you know about this bond? Oh, sure. That's high energy, isn't it? You can use that. So you can actually take an inorganic phosphate and use that to place the coenzyme A. And this enzyme is called phosphoacetyltransferase. Phosphoacetyltransferase. That gives you acetylphosphate. That looks stable? Not particularly. So, can you stick the phosphate somewhere you like on AD? And that will give you acetate. Plus ATP. This is acetate kinase. Acetate kinase is part of a family of enzymes called acyl kinase. Now, you've made acetate. You've made formate. You will also use this to make lactate. And this is called, oh, and then I almost forgot. You then have a enzyme that can take acetyl coenzyme A, and it just reduces it. And if it reduces it, it makes acetaldehyde. That's called aldehyde dehydrogenase. It's an enzyme that's pretty similar to alcohol dehydrogenase. So let's use a different color. So this can also, 2 steps. So we come this will give us the coenzyme A. This is aldehyde dehydrogenase. This is alcohol dehydrogenase. You've made ethanol. You've made lactate. You've made acetate. You've made formate. This is called a mixed acid fermentation. And have you had micro yet? To do a mixed acid methyl red test, this is what you're testing for. You're testing for these mixed acids that drop the pH of the medium. By dropping the pH of the medium, methyl red detects that. So there's more than one way to skin the ethanol cat. But to make ethanol this way, you'll always make it with acid. You'll make it with acetate. You'll make it with formate. You'll make it with lactate, so on and so forth. There is a bacterium that just makes ethanol, and that is the organism Zymomonas. And Zymomonas is used to make, I don't know if you've heard of this stuff. Okay, it's a fermented drink by the Indians. It's pretty bloody strong, and it's used using a bacterium called Zymomonas. You don't make it with yeast, but you make it Zymomonas. I think the Oaxacan Indians are the ones who first made it, if I'm not mistaken. Or was it? Yeah, I think it was Oaxacan Indians. Might have that wrong. But pulque is one thing you can make through this. Okay, that's another fate essentially for pyruvate. Now, let me get to my slide, see where I am here. Okay. Oh, these reactions, they're anaerobic. They're anaerobic. So if you don't have oxygen to perform these reactions, if you do, then these enzymes shut down because you can get more oxygen, more ATP if you use oxygen as a terminal electron acceptor, in which case you will be making ATP via cellular respiration. Now, that's not to say fermentation isn't interesting. Biochemically, it's very interesting, but cellular respiration is the way you make the lion's share of your ATP in cells. Okay, let's talk about how you feed into glycolysis. How you feed into glycolysis. So you've got glucose, and it goes glycolysis. How do you find glucose in nature? It's in polymers, isn't it? So polymers like starch, If you're eating animal tissue, it'll be usually glycogen. Also in our cells, how do you get, well, rather, how is glucose stored in our cells? Glycogen. So how's it broken down? I feel like I'm on candid camera. It's an enzyme called glycogen phosphorylase. Glycogen phosphorylase. Now, glycogen phosphorylase degrades glucose to glucose 1-phosphate. Can that do glycolysis directly? No. You have to convert a glucose 6-phosphate. Fortunately, there is a glucose 6-phosphate mutase. And that will take the phosphate on the number 1 carbon, swing it over to the number 6 carbon. That's an enzyme that gets used a lot in cells. What about starch? We have in our GI tract amylases that degrade starch. Amylases are found in saliva. They're found in the pancreas. They're released into the duodenum. Plants have a starch phosphorylase, which is a very different enzyme that also degrades starch to glucose 1-phosphate. All right, so this is also phosphoglucomutase. Phosphoglucomutase. In fact, it's fair to say that's the more common name. Phosphoglucomutase, PGM. All right, how do you get glucose to go into glycolysis? Remember, you have disaccharides. What do you have to do first? Can you put saccharides in glycolysis? Survey says no. Monomers. You have to convert disaccharides to monomers. So how do you degrade lactose? Lactase, otherwise known as beta-galactosidase. And then sucrose. There's either sucrase, otherwise known as invertase. That's a name for suc sometimes, also known sucrase. Lactose is broken down via lactase, also known as beta-galactosidase. Either one, you can call it either one. I don't. Lactose is broken down to what and what? I don't speak mumble. I heard of glucose. Yeah, glucose is one of them. Galactose, right, galactose. And sucrose, broken down to fructose and glucose. Good. Now, how do these enter glycolysis? Glucose, yeah, and they're done that, got the hat. What about fructose? Well, fortunately, hexokinase isozymes can handle fructose as well as glucose. So hexokinase isozymes A, B, and C can handle both glucose and fructose. What about liver? Because liver has glucokinase, and it can only handle glucose. What do you do then? Do you attack and punt? There is a special enzyme in liver fructose called fructokinase. That's in liver. So that will take ATP, phosphorylate fructose, and phosphorate it to fructose 1-phosphate. Then you have a special aldolase, fructose 1-phosphate aldolase. And that enzyme will disjoin fructose 1-phosphate to dihydroxyacetone phosphate. Plus glyceraldehyde. And then the glyceraldehyde is converted to glyceraldehyde 3-phosphate by an enzyme called triose kinase. Triose kinase. And then you have glyceraldehyde 3-phosphate. And this is converted to glyceraldehyde 3-phosphate by who? Triose phosphate isomerase TPI. So you still get to glyceraldehyde 3-phosphate, you just have a different pathway of getting there. That's the case in the liver. Mannose is phosphorylated by hexokinase, except hexoisomerase handles mannose 6-phosphate very poorly. So there's a very closely related enzyme, phosphomannose isomerase, that will convert mannose 6-phosphate into fructose 6-phosphate. And the bummer is galactose. Let's talk about galactose because it's, out of this whole group, it's the basket case. Sometimes I miss using the blackboard. There's something so satisfying about writing structure on the blackboard. Don't tell Dr. Baldwin I said that. He'll want to come up and hug me. Violate all our COVID rules. Okay, galactose. Sure. All right, triose kinase, mannose, galactose, here we go. So galactose is very common in milk. So being able to metabolize galactose is fairly important. So galactose up, down, yeah. So galactose is phosphorylated on carbon number 1, and that is the enzyme galactokinase. That gives you and that's galactose 1-phosphate. And that reaction should be straightforward enough. It's here that things get a little bit hinky. So now, remember, galactose is an epimer of glucose. But if you want to subject hexoses, well, sugars in general, to some sort of enzymatic modification, you have to have a carrier. That carrier is uridine diphosphate. Uridine diphosphate. So now you're going to take UDP glucose. UDP glucose. And you're going to give this to the galactose, and it's going to come out now with glucose 1-phosphate. And how about over here? Now you're going to end up with galactose that has UDP group attached to it. So this is UDP galactose. Because the isomerase won't make an epimer. The isomerase will take a hexose and make a ketose. That doesn't help you, does it? So to epimerize these, you need to be able to put it on this carrier, because that's your enzymes that handle sugars. That's the binding site they have, is for a UDP carrier. So this enzyme has the most formidable name of UDP glucose. And then after the UDP glucose, you have a pair of semicolon, galactose 1-phosphate, uridylyl transferase. UDP glucose, galactose 1-phosphate, uradilyl transferase. Say that fast 5 times. So this activates the anomeric carbon for transfer to the UDP group. And if you lack this enzyme, you cannot metabolize galactose. You have a genetic disease called galactosemia. Galactosemia. That's a genetic disease that results from a defective gene that encodes this particular enzyme.
Does that enzyme have like certain acronyms?
G1P uridylyl transferase. That's usually, or G1P UTAs. That's usually the one that's used. G1P UTAs. UTAs is pretty common for uridylyl transferase. Okay, now comes the epimerase. So the epimerase is going to take the 6,5,4. It's going to take the number 4 carbon and epimerize it. So this is a epimerase. What kind of epimerase? It's a UDP glucose 4 epimerase. Four because it's the number 4 carbon that it's going to work on. You're going to get and this is now UDP1 glucose. UDP1 glucose. And this is substrate for glycosylation reactions. And you could use it for that. You could hydrolyze this. You just have glucose. You could have it do that too. There's plenty of ways to do that. Of course, that is a high energy bond. So a phosphate can replace that. And UDP glucose synthetase will do just that. It will convert UDP glucose to glucose 1-phosphate. You then give that to glucomutase, and then you're off to the races with glycolysis as well. So lactose is the biggest bummer out of this group. All righty, let's see. We talked about enzymes for degrading sugars, for sucrose, for lactose, for maltose, dextrin, so on and so forth. So, yeah. Now, I'd like to introduce you to a concept called channeling. Channeling. Back in the 90s, channeling was this new age phenomenon where people supposedly had the spirits of some Egyptian gods speak through and so on and so forth. It was all the rage. Do any of you remember the very popular TV show? It was on A&E called Miami Vice. Yeah, Don Johnson, it made Don Johnson's acting career. And then his co-star was Philip Michael Thomas. Philip Michael Thomas was a devotee of one of the more famous channelers. who channeled, her name was J.Z. Knight. She was a news correspondent from Cincinnati or something like that. She would channel this spirit she called Ramsa, and Doonesbury, which is a very irreverent comic strip. I don't know if any of you read Doonesbury. Made endless fun of channeling, and it was quite funny. But anyway, every time I say channeling, I have to put in a disclaimer. We're not talking about somebody getting the spirit of, whatever, and going, glycolysis, whatever. No, channeling was discovered by Barbara Wright and her colleagues when she did something, well, in retrospect, you ask yourself, why didn't anybody else do that? So she took the enzymes for glycolysis and she assayed their kinetics. Then she took the enzymes of glycolysis and combined them. And then did single assays and said, does combining the enzymes affect their velocity? The answer was yes. When you put the enzymes of glycolysis together, their reaction rates went way up. So the enzymes worked faster together than they did singly. Why? Seems as though these enzymes don't float around in the cell, look for each other. Instead, seems as though they're in a complex. So they hand each other substrates. The substrates get to them far faster than they would by diffusion. So the entire process, therefore, goes very, very quickly. And channeling was first found in the enzymes of glycolysis. Then it's been found in several other pathways as well. For example, the urea cycle. Those enzymes definitely undergo channeling. Several biosynthetic pathways, it's clear channeling occurs as well. So this occurs probably more often than you think it would. Also, it's clear that the concentration, the protein concentration, the higher the protein concentration, the more likely these enzymes are to form these complexes. Therefore, the rate, enzyme rate, is dependent on protein concentration of other enzymes that don't catalyze their reaction because those enzymes hand them their substrates. Otherwise, they have to go looking for substrates via diffusion. And that's not quite as fast as having somebody hand it to you. Think of playing tennis, having all those tennis balls to pick up. Imagine what it'd be like if something just fed you the tennis balls, you pick them up a lot faster than doing search and peck around the court to get the tennis balls. That's the best analogy I can think of. Now, the fact of channeling also seems to be a function of where these enzymes are located in the cell, because it's clear that some glycolytic enzymes bind to subcellular structures. For example, it's been known for a long time that hexokinase can bind to the outer membrane of the mitochondria. And every time you isolate mitochondria, you almost always isolate hexokinase stuck to the outside. And hexokinase has a piece at the C-terminus. When you put it around mitochondria, that little piece comes out and it sticks in the outer mitochondrial membrane, and there it sits. So whatever it does, it seems to do near mitochondria. I don't know about you, I think that's kind of cool, but that's just me. Phosphofructokinase and aldolase have the ability to bind to actin filaments. Phosphofructokinase and aldolase, they both bind to actin microfilaments. And then hexokinase binds to mitochondria. Glyceraldehyde 3-phosphate dehydrogenase is known to bind to vesicles. And it might even play a role in vesicle formation. So don't think of these enzymes as just floating around in the cell. They seem to have an address where they live in the cell. And that address puts them all on the same block, to use a city planning analogy. All right, questions about any or all of this?
So is there a
Because mitochondria are known to attach to filaments via myosin. All this works together, doesn't it?
I know.
Yeah. 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 Let's talk about the regulation of glycolysis. Glycolysis is very, very heavily regulated. You'd expect that. Now, there is a process that works in opposition to glycolysis called gluconeogenesis. Yeah, because it makes glucose. 7 of the 10 reactions in glycolysis are reversible, and gluconeogenesis takes 7 steps, runs them the other way, and then for the steps that aren't reversible, it has bypass reactions. So those 10 reactions are used to make glucose. Now, how much sense to have glycolysis and gluconeogenesis working at the same time? It doesn't. This, in biochemistry, is what we call a futile cycle. A futile cycle. Cells work very, very hard to avoid futile cycles. They do it by regulation. So, glycolysis and gluconeogenesis will show reciprocal regulation. What activates glycolysis usually inhibits gluconeogenesis. What inhibits glycolysis tends to activate gluconeogenesis, and so on and so forth. And this actually makes it a little easy to remember. Because if you remember, X activates glycolysis, well, then it probably inhibits gluconeogenesis, and so on and so forth. Now, the steps in glycolysis that are not reversible are step 1, that is catalyzed by kinase. Step 3, that's catalyzed by phosphofructokinase. And step 10, that's catalyzed by pyruvate kinase. Those 3 steps are irreversible. The delta Gs are just too negative. The other 7 steps are reversible. So let's talk about some of those bypass reactions. Let's get rid of galactose here. Because the brain has a relatively absolute requirement for glucose. The brain has a relatively absolute requirement for glucose. If your blood glucose level drops below a certain level, and because the brain takes it up by concentration-dependent means, And organs store their own glycogen. They don't release it except for the liver. But the liver's glycogen stores are limited. So if you want more glucose to ship elsewhere, you need to make it. And so that's why you make it.
So you'll see it from like the rest of the body and the rest of the
Yep. Yeah, many organs will spare glucose for the gain. That's why many of your organs at rest use fatty acids as their preferred carbon and energy source. You ever heard the saying, the more muscle you have, the harder it is for you to gain weight? That's actually true, because resting muscle can use a lot of fatty acids. And those fatty acids won't be stored as adipose tissue. So that's actually true. Ram?
So when you're carb loading, are you taking liver?
Yeah, and you're loading skeletal muscle and you're loading liver with carbohydrates. Now, there have been several studies of carbo loading, and the verdict is it doesn't work. Excuse me, it will give you a 3-day bellyache if you eat enough spaghetti. But the research on carbo loading says it's balderdash. Huh? Yeah, I'm sure it's delicious, but if you want to run fast at a race, don't carbo load. Carbs, too, take up a lot of water, so you'll be pretty bloated the next day. And the other thing they do, from people who do professional races, apparently the trick is to down a lot of caffeine to get your bowels working, to make sure you have a before the race bowel movement. Because one of the worst things you can do is have to go in the middle of a race. And so that's, yeah, I see another athlete over here nodding her head. Carbs, heavy carbs can actually plug you up so that before race bowel movement ain't going to happen. Just a balanced diet. Proteins, fats, carbs, all in about a 30% or so perspective. Also, make sure you get greens, things like that for your joints, things like that. There's no need to carbo load. Nate Martin, who's a graduate here, who wants to run, he wants to make the U.S. team to run the marathon in the Olympics. I've been supporting him on Facebook. Nate's a machine. Have you seen that guy run? He's just a machine. Nate doesn't carbo load. He said if he tried carbo loading once, he got into the 5th mile and he puked. And he almost had the race because his electrolytes were so low. So his advice is don't carbo load. Okay, some of these bypass reactions. So first bypass you'll get if you have fructose 1,6-bisphosphate is you need to then take that phosphate off carbon number 1. And you need to make fructose 6-phosphate, so you're going to phosphatase. And this is called fructose 1,6-bisphosphate phosphatase, or fructose 1,6-bisphosphatase. And this is merely abbreviated FBPase1. That gives you fructose 6-phosphate. And then fructose 6-phosphate is converted into glucose 6-phosphate by phosphohexoisomerase. And if you are a liver cell and you want to release that to the outside world, to the blood, that phosphate will be cleaved off, and this is glucose 6-phosphatase. This enzyme, however, is hormonally controlled. It's only expressed when you have the hormone glucagon. That's why glucagon raises blood sugar levels. It increases gluconeogenesis in the liver, and it increases glucose release by the liver. Yeah. Muscle doesn't have this enzyme. When it comes to glucose, muscles are like the seagulls in Finding Nemo. Remember what they said? Yep, that's what muscle's like. Muscle wants all glucose for itself. Very selfish. Whereas your liver is a more Christian organ, so it shares. Right, we done on time. Okay. No, only use it itself. Pyruvate kinase, there are 2 reactions to bypass that step. We'll get to those one day and show you those 2 reactions. So these 2 are fairly simple. Yeah, Divya? Yeah, you do, and I've been trying to write it, and I keep getting interrupted, and I had a quiz Saturday, and yeah, it didn't go very well. Yeah, I was looking at that. We went up to lipids last time, so the membrane one, I forgot my book. The membrane chapter 13, and chapter 14. Yeah, that's more than enough. 13, 14. No, the membrane one is 10. 10, yeah? Right, we skipped signaling, which is 12. And so it's 11, 13, 14. 11, 13, 14. Okay. Thanks, sorry about that. Just the projector went kablooey. I'm not sure what's wrong with it, but it is what it is. So if they fix it by Wednesday, we'll be back in the saddle. If not, we'll be on the blackboard again. Sorry.
Okay. I will be in lab tomorrow, and then Wednesday is my surgery, so I don't know when that's going to be yet.
Okay.
I might be able to make it to class depending on when it is. I'm not going to plan on that.
Okay.
Anesthesia. I don't know how with it I will be after that. So
You're having general?
yeah.
It's what, shoulder, knee, hip, what?
It's a finger.
Finger, oh. Finger, that shouldn't take long.
No, the surgery they said is 30 minutes. It's going to take longer for just like, getting there and being prepped and then waiting 30 minutes after and everything like
Yeah, no, I think you'll be fine. It's just you'll be in a heap of pain. Your hand will be full of local. Actually, probably won't be till the evening till you start to really feel it.
Right.
Okay. All right.
There's
Okay.
all the recorded lectures online too that I can listen to. So
Yeah, no problem.
all right.
Okay. Ciao.
Bye.